Hi, this is Mrs. Han. I'm going to be explaining how to do the identifying prokaryotic and eukaryotic cells lab. Hopefully you already did the review questions, so you have an idea what the differences between prokaryotic and eukaryotic cells are. Got a little intro to some of the organelles inside, and also watch this quick recap. What I would do is go ahead and pop this out and open it with Cami. And then I would also pop out this answer sheet and open it with Cami so that my links are live. So here I've got my answer sheet. Here I've got the lab. Go ahead and read the scenario. Somebody just came into the ER with kind of some sort of abdominal problem because of water that they drank from a pond. And we're going to go ahead and pretend we're carrying out the lab, but instead of actually setting up everything with microscopes and slides, we're going to watch somebody else do the lab, and then we're going to draw our cells onto this answer sheet. So the first type of cell we're going to look at is prokaryotic cells, and so we're going to look at yogurt under the microscope. So right here, I kind of like to play things at a higher speed, including my own videos. So if you feel like I'm talking too slow, you can always speed it up. But what you want to do is you can sort of watch this person, see what exactly they're what they're getting the sample from. So regular yogurt, if you look, you can see that there's bacteria cultures in there. And then they're going to get a little dip from there, put it on the slide, then look it under the microscope. This is low power, and you can see lots of clumpy stuff, which actually is not the bacteria. It's mostly the protein, and you have to get it a little more focused. So you can see here, there's protein, and these little things are the bacteria. And right here, it kind of gives you some answers, different types of bacteria that are growing in chains. So this is a spear-shaped one. And one of the questions on here is to figure out what type of arrangements that you see. So if you look on the lab, it's asking which of these arrangements do you see? So already this video will tell you that we're seeing Streptococcus thermophilus, which is a sphere-shaped bacteria that grows in chains. So for our table, we can go ahead and we can put that we found streptos chained, arranged round spheres, and then we can go ahead and draw them in a picture. So here the arrangement would be chained or streptos. The bacteria shape would be round or coccus, and then a picture would just be a chain of little circles. And then I'm going to go ahead and just kind of see what else is there. Here's some rod-shaped bacteria. So if you look closely, these are not circles. These are like longer rods, and these are bacillus bacteria, lactobacteria. So we can go ahead and add that we see the rod-shaped bacillus. I'm not sure what the arrangement is, but you can go ahead and just add that that's what we see, the little rod-shaped ones. Okay, and then we're going to go ahead and draw them in different magnifications. So this person went ahead and grew the bacteria so that he could see the colonies a little better under the microscope. And then is going to show them at different magnifications. So this is magnification 400x. So if you look, you can see tiny circles in like a chain-like cluster. So you can go ahead and draw it like that in 400 magnification. I think he then shows it at 1000 magnification, so you can see the Streptococcus bacteria. And then here is the rod-shaped bacteria, Lactobacillus. Okay. And if we go back, we missed the 100 magnification. Here's 100 magnification. Very hard to see, just like, tiny, tiny little dots and whatnot. So that's what you're going to do for part A. You don't have to fill in the whole table because it looks like we only were able to find the bacillus and the coccus bacteria. We didn't see spiral, and most of it was chained. Maybe we saw these in pairs, but I'm not sure. They were sort of, it was hard to see. Okay, and then you're basically going to do the same thing for each type of cell. Now we're going to move on to eukaryotic cells, which hopefully you know have more distinct organelles, including a nucleus. So these are going to be onion cells. And you're going to be drawing them at the different magnifications. So they should get bigger and bigger. And then at 400, you're going to go ahead and label the cell membrane, cell wall, chloroplast, and nucleus. So let's see if we can see what that looks like. It's kind of cool to see how they prepare it, how she prepares it. You can obviously put the sound on for yourself. Just getting a tiny, tiny, thin piece of the onion skin, and she's going to put it into the microscope. She's staining it so that like the nucleus and maybe other organelles might be more visible. So here she just mentioned that this is under 40x. So this would be what you would draw for 40x magnification. You can sort of see these little dots, which are organelles inside the cell. And then she's moving it to 100. So now I can definitely see a nucleus inside. There's the cell wall. They look like little bricks. It's kind of cool. And then she's gonna go a little higher. I might have skipped over it. But anyway, that's what you're going to do for the onion cell. And then you're going to look at some cheek cells under the microscope, which if you were in class, you would just take a little toothpick and scrape the inside of your cheek a little bit and see all the cells. Go ahead and label the 400x ones. This is what, by the way, how they get your DNA. If you're spitting into a vial, your saliva has a bunch of cheek cells, and then they can get the DNA out of the nucleus of your cheek cells. Okay, and then for D, you're going to be looking at pond water, and you're going to see if you can identify some organisms. Here is a chart that's going to help you recognize some of them, and here's another chart. So if you see any of these things, that way you know what it is. And you can go ahead and put that in part D. Okay, and then you're going to go ahead and answer the analysis questions based on your lab. If you have any other questions, you can send me an email and I'd be happy to help you. I hope you have fun.